the first papers about a new phenomenon called CRISPR. In 2007, researchers led by Rodolphe Barangu, who's the editor of one of the journals that I work on, the CRISPR journal, published another landmark paper, this time in Science, that essentially experimentally proved that CRISPR, a region of DNA in bacteria, actually housed a mechanism for precisely targeting the genetic signatures of viruses. And at the time, there were just the first hints that this might be something that could be applied in other settings. The immediate application was in the dairy industry, making cheese and yogurt and all these sorts of things, ensuring that your fermentation proceeded without a virus contaminating that, the big pot of whatever and decimating it was incredibly important. And CRISPR was the key to doing that. So bacteria keep a memory of viruses that they've encountered in these CRISPR repeats and use that information to then recognize many years, generations, eons later, the same virus so that they can recognize it and almost like prime a heat-seeking missile to go after it and destroy the genetic material of that virus before it can land in the cell and destroy the host cell. In 2012, we had the paper that led to the Nobel Prize for CRISPR, a team led by, it was an unusual pairing. They had met just the year before, Jennifer Doudna, an American biochemist, and Emmanuel Charpentier, a French microbiologist. They published a paper in Science, which really got CRISPR to work in a test tube. And they showed that in the same way that bacteria use CRISPR to target a virus and cut the DNA, they could now design CRISPR to target any DNA sequence, which raised literally an infinite array of applications potentially, including the possibility that this could be used in patients, in any higher organism, mammals, plants, what have you.